Hello everyone. In my previous video we talked about the Fred Griffith experiment, that is also known as the transformation experiment, that however proved that non-virulent streptococci they transform to virulent strain, but he unable to justify or prove that this transformation is done by the DNA only and DNA act as a genetic material. Next to Fred Griffith, the renowned work done by the Oswald Avery and his colleague, that is known as Oswald Avery experiment. They continue the organisms that has been used by the Griffith, that is the strain of Streptococcus pneumoniae. But he thought that here Fred Griffith is failed to define the DNA as a genetic material because of two more master molecule may play a role, that is RNA and protein. So they use enzymes, rather the selective enzymes that destroy DNA, RNA and protein, that is the DNAs, RNAs and proteins. So he used this, and for his publication they try to justify that the DNA can act as a genetic material. As figure represents, you can see that they demonstrate that the DNA extract, if not responsible for transformation, then when DNAs will degrade the DNA, that extract should show the transformation. As we can see that the various steps performed by them, in which, just to clarify, in a first step of experiment they try to prepare the extraction or the cell culture of the type R cells, and they observe that no DNA transformation occurs. In the second set of the experiment, they mix the type R cells, that is the non-virulent cells, and type S DNA extract rather than the cells. He mixed it and inoculated and incubated, and he observed DNA transformation. Unlike the Fred Griffith, he didn't use the experimental mice, rather he used the microbiological standard procedures. In third set of practical, he added the same mixture he carried forward as in set of the experiment 2, but he make a change by adding the DNAs. We know that the DNase enzyme will degrade the DNA, and once type S DNA extract we added, DNase will degrade the DNA destroyed, so definitely no transformation occurs. In next set of his experiment, he mixed the same thing, that is the type R cells, type smooth DNA extract, but instead of the enzyme DNase, he used the enzyme RNase. Enzyme RNase will degrade the RNA, but DNA is there, and that's why the DNA transformation observed. In another next set of the experiment, instead of DNase, RNase, he used selectively protease that will degrade the protein, and DNA is not degraded, the DNA transformation occurs. So this way they try to show by using the selective enzymes DNase, RNase and protein that whatever the DNA transformation occurs, that is because of the master molecule DNA, not because of the RNA or protein. In next video we will talk about the another experiment, that is Hershey and Chase bacteriophage experiment. Thank you.